Today we will do PCR experiment, and in here we have 5 ingredients. At first we will add DH2O, double distilled water, inside the solution. Then we will add Biomedical solution, which includes enzyme, buffer and DNTPs inside the solution. And also we will use forward and reverse primers together with the DNA that we want to produce a region of it. Okay, then we are starting the experiment. At first we will take 11.6 microliter of DH2O, Then we will add 15 microliter of Biomedical inside our solution. Then we will add 0.7 from each primer. Between primers, I'm changing the tip in order to prevent contamination of the other primer, okay? Then I'm taking 0.7 microliter from another primer. In order to homogenize the solution, I'm pipetting as well. Then, as a last ingredient, we will add 2 microliter of DNA inside our solution. Alright. Okay, now after adding DNA inside the solution, our solution is ready. At first, we are vortexing the solution in order to homogenize it. Then we are putting it into a PCR machine. In our machine, we have specific PCR program. We are starting with 95 Celsius degree for 3 minutes in order to denature our DNA. Then we will start our each cycle. Each cycle will have denaturation, let's say extension and fine elongation part. For denaturation, again we have 95 Celsius degree for 30 seconds. Then we have 55 Celsius degree for 30 seconds in order to anneal our primers to our sequence. Then we have 72 Celsius degree for 30 seconds in order to extend or new DNA strand. After end of 35 cycle, in here we have final elongation part, which is at 72 Celsius degree, and it will take 1 minute. Today again we will do agrosgel electrophoresis. Again we have 50x tire buffer. That's why we have to prepare 1x tire buffer with 500 milliliter volume. For that we will add 490 milliliter of distilled water inside our graduated cylinder. Then we will add 10 milliliter of tire inside our graduated cylinder. Okay. I'm gonna add 10. Now our 1x tire buffer is ready. Today our agarose percentage is 0.8%. That's why we will measure 2.4 grams of agarose. At first I'm tailing and making 0 gram. Then I'm now adding 2.4. Then I'm gonna add 2.4 grams of agarose inside 300 milliliter of 1x tire buffer. Then I'm gonna shake the solution and I'm gonna put the solution inside the microwave. For 5 minutes at maximum degree. Then I'm putting comb to our mold. Again, we will use these molds in order to prepare gel, in order to polymerizing it. After 5 minutes, we are taking our solution inside microwave. When we put the solution inside, it was like whitish. Now our solution is homogeneous and transparent, but it is too hot to put into our mold. That's why we have to cool it. How? By washing the solution outside. After our solution is cool, we are adding 15 microliter of synth green inside our solution. Okay. Then I'm shaking the solution just before pouring into mold. Then I'm pouring my solution inside the mold. Okay. I'm trying to be sure that there is no bubble which prevents DNA's moving. Then our gel solution is ready to be polymerized into mold. Now our gel is polymerized and we are taking it off from our comb. Then we are putting the gel inside our solution. After putting the gel inside, I'm gonna add 35 microliter of safe green inside the solution as well. Okay, I take it. Then for each solution, I will add 2 microliter of safe green for gel loading. Okay. 2 for leather. 2 for PCR with DNA. 2 for the PCR product without DNA, which is for control, negative control. Then, now I am adding at first 10 microliter of DNA leather inside solution. Then I'm gonna add 10 ml of no DNA solution inside second tube. For our negative control. Then I'm gonna add 10 microliter of all PCR product inside third tube. Then finally, I will insert all the solutions inside our gel. Okay. First, I'm inserting the leather. At first, I'm pipetting in order to get a homogenized solution. Then, I'm injecting the solution into the gel. Then I'm changing the pipette. changing the tip, sorry. Then I'm taking the second solution by pipetting again. Then I'm injecting the second solution inside the valve. Then I'm changing the tip. And then after pipetting, I'm adding the last solution inside the gel. Due to the phosphate backbone, DNA is negatively charged. That's why it will move from negative pole to positive pole. Now I'm going to apply the electricity, apply the voltage. A 150 volts to the gel. Then I will run the gel. After ending for running gel, I'm closing the voltage, and now I'm taking the gel out. The bands, the DNA bands, are only being visualized under UV, that's why now I'm taking the gel out to putting in UV transluminator. After putting gel inside the UV transluminator, we are covering it with filter and we are opening the UV, then we are turning light off in order to visualize our DNA.